This image shows the empty bore of a clinical MRI scanner, the primary physical bottleneck of cognitive neuroscience. Running an experiment requires live human subjects, expensive scanning hours, and the rigid physical constraints of a medical environment. Historically, this friction forced researchers into a divide-and-conquer approach. By using hyper-specific experiments, they mapped isolated functions, like face recognition, to specific localized regions, like the fusiform gyrus. MetaFair is moving away from these fragmented, specialized models toward a unified architecture called Tribe V2. This trimodal foundation model is designed to predict whole-brain fMRI responses across a massive range of conditions. The model was built using over 1,000 hours of fMRI data, recorded from 720 subjects while they viewed movies and listened to podcasts. Because of its training scale, Tribe V2 exhibits zero-shot generalization. It can predict the brain activity of new subjects in response to completely novel stimuli, allowing researchers to run complex cognitive experiments entirely in simulation. This compares human recordings to the model's prediction. The predict row shows the AI estimating brain activity over 12 seconds, tracking the true ground truth above it. The right map shows predicted topography, capturing fine-grained patterns on the cortex as the stimulus changes. Because the AI matches biological geometry so closely in a zero-shot setting, it functions as a reliable digital proxy for empirical research. We can test the reliability of this proxy by focusing on one of the most complex domains of human cognition, the specialized language network. The bottom row shows measured contrasts in the brain when processing complex sentences versus simple word lists, establishing biological baselines. Directly above, Tribe V2 generates predicted contrast maps for the same linguistic conditions. The AI produced these accurate results without task-specific training. The spatial match is highly precise. The simulation correctly predicts the strong lateralization of language processing to the left hemisphere of the brain. These scatter plots chart the correlation between the measured and predicted activity. The model successfully isolates semantic hubs, like the temporoparietal junction, and syntactic regions, like Broca's area, marked here as parcel 45. Replicating these controlled linguistic localizers in silico suggests the model has internalized the actual functional rules of biological brain organization. This diagram on the left of panel B shows the input pipeline. The model does not learn from raw pixels or waveforms. It pulls frozen latent representations from 3 specialized foundation models. It extracts high-dimensional video embeddings using VJEPA2 and audio representations via Wave2Vec-BERT. To capture semantic meaning, it extracts timed text embeddings using Llama 3.2, prepending the previous 1,000 words to maintain linguistic context. Because these models operate at different temporal scales, all 3 are resampled to a 2 Hz frequency. Once aligned, the video, audio, and text embeddings merge into a unified data pipe. This signal enters an 8-layer transformer, aggregating information across rolling context windows. Finally, it downsamples to exactly 1 Hz, matching the repetition time of the fMRI data. This architecture effectively translates abstract machine representations into the rigid temporal constraints of human brain activity. This subject block is the component responsible for mapping those generalized signals to the unique geometry of over 700 individual human brains. During training, the model uses subject dropout. In 10% of cases, it is forced to route through an unseen subject layer, preventing it from overfitting to any single brain and forcing it to learn a generalized human response. This leads to a surprising statistical question. Is an AI's population-level guess more accurate than a recording of a physical human brain? This chart compares individual recordings to the group average. Gray dots are real subjects. The green line is Tribe's prediction. In the Human Connectome project, the AI's prediction is significantly more accurate, doubling the predictive power of the median subject. These results establish Tribe V2 as a mathematically idealized brain, more reliable for identifying population-level patterns than scanning any single individual. Researchers use this idealized brain to run experiments on GPUs. They feed simulated 1-second visual flashes into the model at 8-second intervals. In biological fMRI, blood flow is sluggish. There is a physical constraint called the hemodynamic response function that delays the peak brain signal. This graph of the mean evoked response shows the model replicating that 5-second delay. The activity peaks precisely where it would in a living human subject after the onset of a sentence. Researchers generate their functional localizers by isolating the predicted brain maps at exactly this 5-second peak. By accurately modeling the temporal lag of blood flow, the AI operates as a true mechanistic biological simulator. We can look inside the model using independent component analysis. This reveals what the AI has learned to prioritize in its final mapping layer. Comparing these two rows shows that the model's latent components independently converged on established functional networks. The AI's internal maps in row A align almost perfectly with biological benchmarks, like the visual and language networks in row B. Systematic modality dropout, turning off audio or video input, reveals how different sensory streams merge in the cortex. This cortical map identifies multisensory integration using an RGB color mapping. Red intensity shows text sensitivity, green shows audio, and blue shows video. The color mixing identifies bimodal zones. For example, yellow regions in the temporal lobe identify areas where the model's text and audio representations fuse together. The model reveals the underlying organizational logic of how the human brain processes a multimodal world. Current limitations remain. fMRI lacks millisecond-level resolution, and the model treats the brain as a passive observer rather than an active agent. This chart tracks the model's scaling laws. As more training data is added per subject, the encoding accuracy continues to rise log linearly without any visible plateau. Tribe V2 establishes a scalable digital proxy of the human brain, a unified platform for the next era of in silico neuroscientific discovery.